بسم الله الرحمن الرحيم. الحمد لله رب العالمين والصلاة والسلام على رسول الله الأمين. مرحبا يا أبنائي وبناتي في أكاديمية السودان للعلوم الطبية الافتراضية. وأنا أولا أعتذر جدا للتأخير في الجلسة دي، وأتمنى إنه نبدأ الآن ببداية جيدة وإن شاء الله يتعوضوا في المرات الجاية. My name is Dr.Mohammed Ohaj Mohammed, Associate Professor of Biochemistry in University of Bisha and Medical Education as well, and previously I was in University of Jazeera. I will start this session straightforward by what I have prepared in my presentation is only 3 objectives main. As you can see, the first objective to explain the primary, secondary, and tertiary structure and quaternary structure of the protein. The second objective, describe the factors affecting the 3-dimensional structure of the protein, and describe the protein denaturation. Between these, I will give some extra elaboration and explanation about some of the aspects of the protein functions and the classification of the protein. So if you can see my presentation and the PowerPoint, I made it in a way to be like some sort of animation that can let you follow easily. So as introduction, I think we all know that the proteins are most abundant molecules in the living system, whether in the structural parts of the body, in the muscles, as the cells, as the tissues. In everywhere you find the proteins, and or inside the individual cells as type of the constituent of the cell. Also, every life process depends in a way or another in the proteins. If we take, for example, the digestive system, the process of digestion, the absorption, and the different pathways of metabolism depends mainly on the proteins, whether as enzymes or as hormones or as mediators of the different processes. In every aspect of the life, you find the proteins play a very important role in the living cells or the tissues or the organs as well. All proteins, as you know, from chemistry point of view, are the polymers of their individual amino acids, which are linked together by the covalent bond known as peptide bonds. So every protein is, in general, chemically defined as a polypeptide chain of proteins which are linked by peptide bonds mainly, and sometimes you can find some other bonds like the sulfur bridges or the disulfide bonds. Just to make things easier, I make like introduction to what are the amino acids from the structure and how they are linked into groups to make the peptide bonds. So this animation, if you can see, this is the general structure of the amino acids, where you have the carboxyl group in the black and the amino group in the green, yes, and the H in the hydrogen atom in the blue box, and the R in the red box, which all together attach to the carbon atom, which is known as the alpha carbon atom. So this is the general structure of the amino acid. This is alpha amino acid, and this amino group, this is a carboxyl group, the side chain, the R distinctive group, and you know that, I think, from maybe your previous study, that amino acids differ in the structure of the R side chain or the R distinctive group. Why we call it alpha amino acid? Because you know that the carbon atom which is adjacent to the carboxyl group, the main group in the functional group of the amino acid or the fatty acid even, is known as alpha, and this is the alpha amino carbon atom, where we have the other substituents are attached to this alpha carbon atom. In nature, you can find some of the amino acids attached to the beta carbon atom, which is the second carbon, or very rarely you can find some to the gamma. This is the last group here. Carboxyl group is a side chain which containing a carboxyl group, and this distinguishes the different amino acids in the living cells, which are almost all or are of the alpha amino acid. You know also the amino acids have got typical structure like the carbohydrates, that they can have some optical activity in rotating the plane polarized right either to the right, and there we know as the amino acid, or against or opposite to the right or to the left, rotatory, and they are known as LL amino acids. And since all these constituents are related to the alpha carbon, so we can know the amino acid as L-alpha amino acid or D-alpha amino acid. In our living cells, especially the human, the most dominant are the L-alpha amino acid. According to the position of the amine group, whether it's to the right of the asymmetric carbon or to the left, the amino acids are classified into alpha or into D or L amino acids. The peptide bond is that, before going to the peptide bond, I'm not sure whether you know the amino acid and their classification. In nature, we have got different type of amino acids in general. We can have up to more than 300 amino acids in nature, but they are not part of the protein. They can be found as plant toxins or some bacterial toxins. Another class of the amino acid is the 20 amino acids, which are known to be encoded by the genetic code in the DNA, specific coding for the protein synthesis. And these 20 amino acids are the most dominant in our living cells as part of the polypeptide of the proteins, enzymes, and some hormones, and some other type of the molecular constituent of the body. The third class is that which is type of the 20 amino acid, but they have underwent some type of modification. They are known as post-modified amino acids. So these post-modified amino acids, they will be, during the preparation of the polypeptide chain, they can go into some interactions where they can attach some functional groups like the hydroxy group, for example, or remove some groups or remove some of the constituents of the amino acid. Then it will be known as post-transitionally modified amino acid. So in this modified form, it's not part of the genetic code or not encoded in that final form, but it is originally one of the 20 amino acids which are coded by the DNA genetic code. The 20 amino acids which are encoded by the DNA, they are classified into 5 or 6 groups according to the specific structure of the R side chain. So you can find some amino acids containing an aliphatic group where the R is only carbon and hydrogen, or only hydrogen, like the case of the glycine. Or you may find some of the amino acids contain CH2, CH3, for example, like the alanine, CH3 only, and so on. Or the R side chain may contain, besides the aliphatic group, some functional group like the hydroxyl group. Or you can find the R side chain containing a carboxyl group, or an amino group, or a sulfur bond which can form the sulfur bridges. So this general review of the structure of the amino acids and classification, but they all share the general structure where we have a functional group, the carbon atom, the alpha carbon atom, which contain the carboxyl group, the amine group, and the hydrogen with the R distinctive group. So the peptide bond is known as the bond which link two amino acids by removing a water molecule or dehydration condensation, and the linkage is between the carboxyl group of the first amino acid and the nitrogen or the amine group of the second amino acid. And this type of bonds, because there will be a water molecule removed and replaced by this peptide bond, is known as the peptide bond, or sometimes you can find in some references as the amide bond, because the bond takes place between the carboxyl group of the first amino acid and the amine group of the second amino acid by removal of water, and it's known as dehydration synthesis. So this peptide bond is a type of the covalent bond, because the carboxyl carbon and the amine nitrogen will share their electrons to be stable. So because of this sharing, they are known as covalent bonds. The biochemically important peptides include the following list. If you can see my presentation, the insulin, for example, contain 2 chains of the total of 51 amino acids. The glucagon, both which are secreted by the pancreas of the body, also contain 29 amino acids, polypeptide chain with about 29 amino acids, while the ACTH contains 39 amino acids, and the thyroid hormone contains 3 amino acids only, tripeptide, and the oxytocin contains 9 amino acids, and the antidiuretic hormone contains 9 amino acids, and the glutathione contains 3 amino acids. Shabab, can you hear me clearly?
Yes.
Okay, any questions so far? Do you have background about the amino acid and the peptide chain, peptide bonds, and the polypeptide chains? Do you think that we need some more elaboration on that, amino acid and the peptides? Any reaction for Shabab? You can unmute yourself and communicate. ها شباب، سامعيني بصورة جيدة يا شباب؟ هل البرزنتيشن بتاعي في السكرين اللي انا عارضها واضحه عندكم ولا قاعد تتابع من اجهزتكم الخاصه؟ عاوز تفعل يا شباب؟ نعم؟ واضح السكرين بتاعتي انا ولا بتاعتكم انتم شغالين من اجهزتكم الخاصه؟ يعني انا شغال في بتاعت البيولوجيكال امبورتنت بيبتايدز في السكرين، واضحه عندكم المجموعه دي؟
ايوه واضحه يا دكتور.
فاين جميل. طيب نواصل. يبقى يا شباب الامينو اسيد او البوليبيبتايد تشين بتكون من 3 احماض امينيه بنسميها تراي بيبتايد. اذا كان بتكون من 4 زي ما في حتى الجلوتاثيون تراي بيبتايد، او الثيرويد هرمون تراي بيبتايد لانه بيكون من 3 احماض امينيه. والانتي ديوريتيك هرمون فور اكزامبل بيسميه نونا بيبتايد او اوليجو بيبتايد. فاذا الفكره الاحماض الامينيه والكلاسيفيكيشن بتاعها واضح، والخصوصيه دي انه كل حمض اميني يعني يكون مختص ب R side chain distinctive بالنسبه له، بتميز عن باقي الاحماض الامينيه. الحكايه دي واضحه، والبيبتايد بوند بتكون كيف هنمشي قدام في الامثله بتاعت هاو البروتين ستراكشر تاخذ شكلها النهائي ثلاثي الابعاد. اوكي أواصل؟
تمام يا دكتور.
So the protein in general is a linear molecule of polypeptide chains that can contain thousands of amino acids, and all these amino acids basically are connected by the peptide bond. between each amino acid and the other. You can imagine a polypeptide chain of a protein containing some thousands of amino acids in the chain, can be only a single chain, or you can sometimes find multiple chains and they can be linked by another way of covalent bond, like the case of the insulin, which we'll take an example of it in the subsequent slides, that there are two separate chains, but they are linked by the disulfide bonds or sulfide bridges. But usually this linear chain of the amino acids, which are connected either by peptide bonds or the disulfide bridges, it's not the final structure of the protein, and it will not do any function of the protein unless the protein rearranges itself in a three-dimensional conformation or structure. And at this stage, the protein is known as a native protein. For example, the albumin is one of the proteins of the plasma. It has got a very specific three-dimensional structure which is unique to the albumin. So it will not reach to be a fully native albumin protein unless it has its unique three-dimensional structure, and this will take place by the folding of the basic chain, whether a single or multiple chains. So generally, the structure of protein is studied into 4 levels: the primary structure, secondary structure, the tertiary structure, the quaternary structure. And the primary structure is mainly related to the skeleton of the amino acid of the protein, which is the polypeptide chain of the amino acid, which is encoded from the genetic code and being translated or transcribed by the RNA. And this is the level where you have only either the peptide bonds in the main skeleton linkage, or sometimes the disulfide bonds. The secondary structure of the protein, where you have some relationship between either the same chain, the polypeptide chain of the amino acids of the protein, by forming some other non-covalent bonds, like hydrogen bonds, for example, or van der Waals forces, or ion-ion interaction forces, or electrostatic interactions. And in this case, we'll find some details later. The tertiary structure is where the multiple polypeptide chains, which they have formed the primary structure and secondary structure, they will be linked into an extra level of forming the primary structure of the three-dimensional structure, or in other words, where it will take its final shape or semi-final shape into three-dimensional structure, where each alpha helix or beta sheets, which will be explained in the subsequent slides, will join by, again, non-covalent bonds. And then the quaternary structure, this is a higher order of the protein, and especially for some types of enzymes and some type of the plasma protein like the hemoglobin. They can go up to the quaternary structure, and this structure, or this level of the protein structure, is where different polypeptide chains, 2 or more, can have some sort of interaction forces, like hydrogen bonds, van der Waals forces, or electrostatic interaction between the polypeptide chain A and, for example, B, or A and B and C. So you can have more than one polypeptide chain, each with three-dimensional structure, where it has gone through the primary, secondary, and tertiary structure. So, in general, the protein structure is studied into these 4 levels. The primary structure, it is where the different amino acids are linked into the linear chain by either polypeptide chains or some sulfur bridges. And at this level, we are talking about a covalent bond. This covalent bond can be a peptide bond or a sulfur bridge or disulfide bond between two cysteines or cysteine methionine. The secondary structure of the protein is where either the same polypeptide chain will have some sort of relation between the functional group of amino acid number 1 and amino acid, for example, number 4, by forming hydrogen bonds, and the second amino acid with the fifth amino acid, and so on, up to the end of the chain. You can find this alpha helix, where the protein, the polypeptide chain, will form itself like the strand of the DNA in alpha helix shape. المستوى ده يا شباب من تكوين البروتين بيكون فيه سلسلة الأحماض الأمينية الأساسية، اللينير تشين، الكون بواسطة الجينيتيك كود. بتكون في علاقات بين كل حمض أميني والحامض الرابع تقريبا منه. بيكون في علاقة بتاعت هيدروجين بوند بين الكربوكسيل جروب بتاعت الحامض الأول فور اكزامبل، ولا الهيدروجين بتاعت الحامض الأول والكربوكسيل بتاعت الحامض الرابع. فبتعمل نفسها في شكل حلزوني بتاع الألفا هيليكس دي في اليسار هنا، والصورة في اليمين بتورينا نوع ثاني من السكندري ستراكشر. again هي عبارة عن هيدروجين بونز بصورة أساسية، وبتكون بين two adjacent polypeptide chains، سلسلتين من الأحماض الأمينية مرتبطة كل واحدة فيها ببيبتايد بوند فيما بينها، وبيكونوا قريبات من بعض. بتحصل نوع من الانتراكشن فورسز بين الكربوكسيل جروب بتاع حامض معين في سلسلة A فور اكزامبل مع الهيدروجين بتاع النيتروجين بتاع السلسلة الثانية، بيدينا ما يسميه بالبيتا بليت الشيش في الشكل بتاع الزجزاج. فالخلاصة في المستوى ده يا شباب من تكوين البروتين إنه إما السلسلة نفسها تعمل إعادة تشكيل لنفسها في شكل يعني ثنائي الأبعاد، بحيث إنه يكون فيها يعني شكل حلزون بتاع ألفا، أو شكل بتاع زجزاج بتاع بيتا. الألفا بتكون ألفا هيليكس بين نفس مكونات السلسلة الواحدة، والبيتا شيت إما يعني two separate polypeptide chains، أو البيبتايد chain الواحدة بعد ما يحصل لها فولدنج تنطبق على نفسها، ويكون في الأحماض الأمينية في سلسلتين قريبات من بعض بحيث يعملوا hydrogen bonds. So we are talking about chemically the hydrogen bonds in both these alpha helix and beta pleated sheets. المستوى الثالث من المستوى بتاع تكوين البروتين structure، it is where we have multiple alpha helix and beta sheets joined together by non-covalent bonds forming the three-dimensional structure of the protein. And this is a polypeptide chain. Can be a single polypeptide chain, or you may have two polypeptide chains. Sorry. They are joined to each other by non-covalent bonds. Can be hydrogen bonds, can be electrostatic attraction, can be van der Waals forces, or hydrophobic forces between the different parts. And at this level, we are talking mainly about the forces which can be influenced by the nature of the R side chain, because all the main functional groups, they have been incorporated into either the alpha helix or the beta sheet of the secondary structure. بالعربي كده شباب، المستوى الثالث من تكوين البروتين او يعني تركيب البروتين بتكون فيه علاقات بتاعت روابط غير تساهميه بين مجموعه بتاعت الفا هيليكس مع بيتا شيت مجاوره، او سلسلتين الفا هيليكس ومعاهم واحده بيتا شيت، او ما العكس يعني، فبحيث انه يكون في اكثر من شكل سلسله الفا هيليكس وبيتا شيت زي ما الموضوع واضح عندكم هنا في الريكتانجلر ده، في الفا هيليكس هنا وجنبها في مجموعه ثانيه مجموعات متوازيه مع بعض تدينا بيتا شيتس. فالعلاقات ممكن تحصل بيناتهم بحيث انه يكون فيها روابط وانتراكشن بيناتهم، ما هي تكون كوفيلنت بوند، ما فيها مشاركه على الالكترونات، بانما هي جس من نوع من التجاذب، والقوة بتاعت الربط بيناتهم وهي الهيدروجين بونز او الفان دير فالز فورسز او الايونيك بونز اللي يسميها الكتروستاتيك فورسز. ده المستوى الثالث من تكوين البروتين، وهنا يكون البروتين اخذ شكله ثلاثي الابعاد 3 dimensional structure of the protein. The quaternary structure, which is as I said, it's a higher level of the protein structure, where you have multiple polypeptide chains. It can be 2, 3, 4, or even more than that. Each one has its own three-dimensional structure, and this three-dimensional structure have been achieved, or the polypeptide chain has reached that level of the three-dimensional structure by having the main skeleton of the polypeptide chain, the primary structure, and the secondary structure with alpha helix and beta sheets, multiple of them, and then they form their relationship by non-covalent bonds, forming the tertiary structure. So you can imagine the hemoglobin in this image, where you have got 4 units, 2 of alpha globulin and 2 of beta globulin, joined to each other by non-covalent bonds, and these interaction forces can hold the polypeptide chains together. So this level is known as the quaternary structure of the protein. Again, the primary structure يا شباب شوي من التفصيل. It is the primary structure which is held by mainly peptide bonds, and you can find some sulfur bridges. Regarding this structure, the primary structure, you can imagine this polypeptide chain, and you can look at the animation which will show you which amino acid will start the reaction. Usually will start from the the left side, in this case for example, and this first amino acid has its carboxyl group, will lose or lost its hydroxyl group, and the nitrogen of the second amino acid hydrogen, and they join this peptide bond where the arrow is pointing. So because in the first amino acid with the green highlighting has a free nitrogen end, or an amine end, is known as the N terminal of the polypeptide chain, and on the other side you will find a free carboxyl group which has not entered into any bond, is known as the C terminal, or the carboxyl terminal side of the polypeptide chain. So these two amino acids, the amino acid number 1 and amino acid number 2, reacted by removal of water molecule between these two amino acids, and they form the peptide bond number 1, which is shared by or highlighted by the arrow. Then the dipeptide with the shared amino acid is a yellow highlight. Also the link to these by a peptide bond where the arrow is showing. Then the one with the pink color, and the other more clear, and finally the N terminus, like you can name it as the red color circle. So in general, the polypeptide chain in the primary structure of the protein has this structure, and between each two amino acids there water molecule have been removed, and this is why we call it the condensation synthesis or the dehydration synthesis. Condensation or dehydration synthesis, where we remove water molecule, as I told you, the hydroxyl group from the first amino acid, and the amine group, the hydrogen from the amine group with the other amino acids. So by this way, you can also easily recognize that the amino acid has 2 ends, either an N terminal or a C terminal. N refer to the amino 3 amino acid amino group terminal, or the carboxyl terminus or terminal, where you have 3 carboxyl group which has not entered into any peptide bond. Can you see my pointer? I tried to use a pointer with this light pointer. شباب شايفين البوينتر ده؟
واضح.
في مؤشر وين؟ في الحمض الأمين الأول والثاني. إيه؟ So this is peptide bond number 1, peptide bond number 2, peptide bond number 3, and peptide bond number 4, and peptide bond number 5. So these 6 amino acids have been linked by 5 peptide bonds, and the direction of the polypeptide chain will be from the N terminus to the C terminal, And this is very important to understand the reactions of the proteins, the secondary structure and the tertiary structure in the subsequent slides. So the primary structure is related to the number of amino acids, the sequence of the amino acids, and the frequency of the repetition of the amino acids. For example, you can have in the polypeptide chain of 100, for example, 46, like the case of the sickle cell anemia, for example. You can have some of the amino acids repeated in some way of frequency, or something they can be either like this case, for example, the TYR refers to tyrosine, GLY for glycine, and PHE for phenylalanine, and LUE for the leucine. This is a polypeptide chain of 5 amino acids, and again this is another polypeptide chain of 5 amino acids. The bond between each of these amino acids is peptide bond, as you know, and the only difference between these two in the last amino acid where you have the leucine in the first polypeptide chain and the methionine in the second polypeptide chain. So the first one is known as leucine enkephalin, and the second one is known as methionine enkephalin. So these are two polypeptide chains almost similar in every aspect, only different in the last amino acid. So these are two polypeptide chains. The bond between these are peptide bonds. This is the structure of the insulin, as I mentioned in the slide. Contains in total 2 chains A and chain B. The total is 51 amino acids. So each of these circles stands for single amino acid, and this polypeptide chain A contains this group of the 21 amino acids, and the chain B contains 30 amino acids in the other color. If you can see my pointer here, I'm pointing to the sulfur bridges. Wherever you have a cysteine and a cysteine in the other polypeptide chain, they can form the sulfur bridges, which can link these two chains, and the other one is cysteine, and the other cysteine also forms a sulfur or disulfide bond, also known as the sulfur bridges, because it links between the two polypeptide chains. This sulfur bridges can be between the two different chains or within the same chain after folding. You can imagine this as one single polypeptide chain, and because instead of having this carboxyl group, you can imagine this continue up to the end of this carboxyl group, and it has been turned around, like folded around. يعني إذا كان في سلسلة واحدة طويلة وحصل لها انطبقت على نفسها أو حصل لها folding في اتجاه النفس بتاع جاية منه، بيحصل إنه أي two cysteines جو على مسافة قريبة من بعض ممكن يعملوا sulfur bridges يقووا العلاقة بين التو bonds. ممكن السلفر بريدج كذلك تكون في نفس السلسلة زي ما في الحالة بتاعت التو اس الموجودات فوق ديل. Again, this is type of sulfur bridges, disulfide bond between the cysteine and the cysteine in number 6 and number 11. السلسلة دي يا شباب ممكن تخيلها لأي بروتين ثاني، بس هنا عندنا عدد محدود، لكن هناك في حتى البروتينز تكون السلاسل طويلة جدا، ممكن تكون بالآلاف. عادة عشان نسمي البوليبتايد chain بأنها polypeptide chain of a protein, usually it should be around 1000 kilodalton or more. But you can find some amino polypeptide chains of proteins with less than that molecular weight, but the general proteins have got very high molecular weights. Okay? Any questions so far, Shabab? إسراء أحمد تابع يدك ولا ظاهر في السكرين؟
أي سامع ظاهر جيد، ما في بداية أسئلة.
أوكي، في سؤال؟ طيب إذا ما في خلونا نمشي للسكندري ستراكشر أو قبلها نشوف أهمية البرايمري ستراكشر. نعم تفضلي. في السلايد السابق
الدايسولفايد بوند.
ايوه
الدايسولفاين بتكون بس بين السيستين امينو اسيد يعني؟
اذا في تو سيستين قريبات من بعض لانهم فيهم في كل سيستين عنده سلفر بون او سلفر اتوم معاه هيدروجين، يحصل دي هيدروجينيشن يدونا السلفر بريدج في الحاله في انا مشيلها هنا، او في سيستين والسيستين هنا الثاني. اذا في تو ميثيونينز قريبات لبعض الامينو اسيد تحتوي على السلفر هي السيستين والميثيونين. فاذا كان في سيستين او ميثيونين وميثيونين او ميثيونين وسيستين ممكن يكون عندهم فرصه يعملوا سلفر بريدجز او سلفر دايسلفايد بوند. تمام؟ whether between the same chain زي ما في الحاله اللي فوق دي تعمل يعني strengthening للبوندز بتاعت البيبتايد، او تعمل لينكينج للتو بوليبتايد تشينز المختلفات بصوره كامله. اي سؤال ثاني؟ اي سؤال ثاني يا شباب؟ السؤال الاول ده كان من رهف ولا اسراء؟
ايوه يا دكتور.
استاذتي
رهف.
ايوه جميل. طيب اي سؤال ثاني يا شباب؟ حتى الان كلامي كله عن البرايمري ستراكشر يا شباب واهميتها. قلنا تذكروا في حاجتين اساسيات، ان هي الهيكل الاساسي بتاع البوليبتايد تشين بتاعت البروتين بتتكون من كوفالنت بوند، اما تكون بيبتايد زي ما في الحاله بتاعت التو امينو اسيد بوليبتايد تشينز الفوق ديل، او تكون سلفر بريدجز زي في الحاله بتاعت الانسولين السلفر بريدج الرابطه بين التو تشينز A and B في موضعين، او مقويه السلسله بتاعت A بين تو سيستينز. الحالتين سواء كان بيبتايد بونز في السلسله الاساسيه بتاعت A او B او السلفر بونز دي كلهم عباره عن كوفالنت بونز في مشاركه الالكترونات. اوكي؟ المسارات الثانيه مختلفه شويه بروابط غير تساهميه. طيب من اهميه البرايمري ستراكشر انه determines الاذر ليفلز، يعني بيعتمد المستويات الثانيه من تكوين البروتين سواء كان المستوى الثاني او الثالث او الرابع، كلها تعتمد على طبيعه البرايمري ستراكشر بتاعت البروتين في البرايمري ستراكشر في اللينير تشين بتاعت الامينو اسيد. بمعنى اخر اللينير تشين بتاعت الامينو اسيد دي هي بتحدد انه هي مجموعه من الاحماض الامينيه او السلسله حتمشي بشكل الفا هيليكس في زي ما هشوف بعد شويه، وهي مجموعه منها حتعمل في شكل بيتا شيتس، و200 حيعملوا كومبينيشن بين 2 الفا هيليكس او 3 الفا هيليكس معاهم بيناتهم واحده بيتا شيت فور اكزامبل. Also a single amino acid إذا كان substituted in any polypeptide chain of the primary structure by any other amino acid, this can be raised to malfunction of the protein, like the case of the following example. Look at this chain. This is a beta chain of the 146 amino acids. The first one is VAL, stands for valine, histidine, leucine, threonine, proline, glutamine, and glutamine. Okay? Or glutamic acid. And this is a normal beta chain of the hemoglobin, and it is up to 146 amino acids. يعني سلسلة بتاعت الهيموجلوبين البيتا تشين بتكون من 146 حمض اميني. دي الاول احماض امينيه في السلسله من الناحيه بتاعت الان تيرمينس. اوكي؟ والناحيه النهايه الامينيه للمركب. So look at this and look at the second example of the beta chain. Again, it's the same beta chain with the same set of amino acids: valine, histidine, leucine, threonine, proline, valine, and glutamine or glutamic acid. So which one is, what's the difference between these two? واحد من الشباب متابع يا شباب؟ أيوة إسراء.
In second chain, glutamine is converted by valine.
Valine. So the substitution of the glutamine with valine makes this polypeptide chain slightly different because the glutamine have been replaced or substituted by the valine. And this is the abnormal beta chain of the hemoglobin, which will lead to the sickle cell RBCs or sickle cell hemoglobin, and this will lead to the consequences of the sickle cell anemia and the drawback of all the side chains, the symptoms, the complications of the patient, only because of replacement of one single amino acid in the polypeptide chain of the beta chain of the hemoglobin. So the primary structure is very important in the number of amino acids, the way of the arrangement, the sequence where they are arranged, and the proportionality of the amino acid. For example, in this one we have got 2 glutamine, and 1 threonine, 1 proline, 1 leucine, 1 cytidine, and 1 valine. While in the other one we have got 2 valine in different positions, and we have the other as you can see it. So the substitution of only single can make this effect of the sickle cell anemia, and it is all complications. If you can imagine if you replace more than 1 amino acid, that means it will be completely malfunctioning protein, or it will not do its function as expected. So this glutamine has been substituted by the valine in the abnormal beta chain of the sickle cell patients or their blood, and this will lead to all the complications. Secondary structure, it is mainly related to the alpha helix in the top. I presented two different types of the alpha helix, or 2 different orientations, and the beta sheets in the bottom. So the alpha helix and beta sheets, as I mentioned before, it is known as the secondary structure of the protein, and this is mainly about the non-covalent bonding that can solidify the structure of the protein. So the alpha helix is due to the conformational or configurational relationship between the residues within the same polypeptide chains of the linear sequence of the primary structure, usually between the 3rd or the 4th amino acid, between each amino acid and its 3rd or 4th amino acid. يعني بين كل حمض اميني والثالث منه او الرابع بتحصل الالفا هيليكس دي بهيدروجين bonding. This will give us the twisted shape of the alpha helix, الشكل الحلزوني بتاع alpha helix. هنا في الاخضر في البوكس الاسود ده بدي الشكل العام بتاعها. هنا موضح الاكسس الاساسي بتاعها، والنقاط الزرقاء او الخضراء دي عباره عن الريزيدز بتاعت احماض امينيه. فالنقاط الصغيره لو شايفينها بين كل واحده وهنا في الصوره الرئيسيه دي الهيدروجين بونز بين اذا اعتبرنا ده الامينو اسيد رقم 1 2 3 4 بين 1 و4 فيها هيدروجين بونز، بين 2 و5 فيها هيدروجين بونز and so on. So بين each amino acid and the 3rd or the 4th amino acid you can have hydrogen bond between the functional groups, or beta sheet between 2 adjacent or close polypeptide chains, or the same chain after being folded back to itself. So this is the beta sheets. So we are talking about the hydrogen bonds which is known as non-covalent bonds, because there is no sharing of electrons, only weak interaction between slightly positively charged hydrogen attached to the more electronegative nitrogen and slightly negatively charged oxygen of the carboxyl. The secondary structure level is mainly about the non-covalent bonds, can be hydrogen bonds, electrostatic ions or bonds, the ionic bonds, or hydrophobic interactions, or the van der Waals forces. But mainly in the primary and secondary structure, it's hydrogen bonds between the functional group of either the same chain or 2 adjacent chains. The other bonds can be found in the secondary structure, but with less importance or less influence in the stabilization of the secondary structures, alpha helix or the beta sheets. The tertiary structure is where you have combination between different alpha helix, for example, alpha helix number 1, alpha helix number 2, to the left. These two polypeptide chains, secondary structures, the alpha helix, have been combined or attracted by this ionic bond between the negatively charged ion of the positively charged ion, the red color, and the negatively charged ion or the other side, the white colored ball. So this is ionic bond. It is a type of electrostatic non-covalent bond. Or some polar forces where you have some carboxyl group, for example, and amino group, which is some positive and some are negative, and they can have some type of the ionic bonds, or sorry, polar forces, or some water molecules around the protein polypeptide chain. These lines stand for the beta sheets. So the beta sheet at this point has been attached to the alpha helix by some non-covalent bonds, and this also beta sheet with this polypeptide alpha helix have been attached by these sulfur bridges, and these are type of non-covalent bonding that can form the three-dimensional structure of the protein. So the three-dimensional structure is denoted by the tertiary structure of the protein, where the combination of some alpha helix and beta sheets can have some other non-covalent bonds holding them to make it more stable and to have the thermodynamically the most stable structural configuration of the protein. The arrangement is known as protein folding, where the different part of the protein in the linear chain, after forming the alpha helix and the beta sheets, they can come together by some non-covalent bonds forming the three-dimensional structure of the protein. Again, chemically the bonds here can be either hydrogen bonds or electrostatic ionic bonds, or hydrophobic interactions, or van der Waals forces. And all these together shows that they are non-covalent forces or non-covalent bonding. There's no any sharing of electrons in any of these. The other point to be mentioned in this level is that the tertiary structure is usually between the R side chains of the amino acid, not between the functional group of the main amino acid, because all of the functional group have been incorporated either into the alpha helix or some of the beta sheets. So the majority of the bonds here, whether hydrogen bonds or electrostatic ionic bonds or hydrophobic interaction or van der Waals forces, are between the R side chain of an amino acid in one of these groups and the R side chain of the other group, and so on. So these are mainly about the R side chain nature, which can make this three-dimensional structure. The quaternary structure of the protein is known as I mentioned, the higher level, where we have different polypeptide chain, each which has its own primary structure, secondary structure, and tertiary structure, has a fully oriented three-dimensional structure, and they came together into some sort of interaction, and they hold by non-covalent bonding. So certain polypeptide chains, as I mentioned here in this box, will be aggregated to form one functional protein. Can be 2, 3, 4, or more. Again, the bonds between these polypeptide chains are again non-covalent forces or non-covalent bondings. Can be hydrogen bonds, electrostatic bonds, hydrophobic interactions, or van der Waals forces. Depending on the number of the polypeptide chain of the protein in the quaternary structure, it can be named as a dimer, where it contains 2 polypeptide chain, each with its tertiary structure, or it can be a monomer, the same polypeptide chain of the protein with a very long chain. It can form 2 or separate different units, and they came together into one functional group. Or it can be known as trimer or tetramer, where it can contain 4 chains. And this is an example of the hemoglobin, where 4 separate polypeptide chain, each has this 3-dimensional structure, passes the primary structure, secondary structure, and tertiary structure. They came together, aggregate to form the quaternary structure of the hemoglobin, as we see in the example. So the protein will lose its nature if this orientation or this aggregation by any means it has been dissociated or separated. So the subunit, if they dissociate by any means, that means the quaternary structure is not anymore valid or not anymore like present. So the protein will lose its function because some of the units have been separated or dissociated apart from their other units. So this is 2 examples. This is a dimer where this is the pink color has got a fully oriented polypeptide chain with alpha helix, beta sheets, and the primary structure already there. So it has got a 3-dimensional structure, and it came closer to another polypeptide chain in the green with again 3-dimensional structure, and they form some type of interaction forces in this joining edge or joining position. It can be hydrogen bonds, or can be van der Waals forces, can be ionic interaction, or can be hydrophobic interaction between the constituent of these 2 polypeptide chain. So what is different in this structure is that we have got each of these polypeptide chain completely has its 3-dimensional structure, which is unique to it. And it came in close contact with another group of polypeptide chain, each with its 3-dimensional structure, and they form this interaction. And this is a dimer because it contains only 2 units, each with its complete 3-dimensional structure. The other most commonly used example is that of the hemoglobin, where we have got 2 beta polypeptide chain, each with its 3-dimensional structure. And wherever you find this type of rod shape, it means alpha helix. And wherever you have found some closer line to a parallel, that means beta sheets. So this is polypeptide chain beta, and the other polypeptide chain beta, each with its 3-dimensional structure. They came together by some sort of forces that hold this dimer, and again these other 2 polypeptide chain alpha. Again, they have got some type of forces, they hold each other, and between each alpha and beta, again, there are some type of forces. So all these forces which hold the quaternary structure of the protein between the 2 beta and the 2 alpha, either between the same polypeptide chain or within the different polypeptide chain, all are none type of these, either hydrogen bonds, electrostatic or bonds, or hydrophobic attractions or van der Waals forces. So this is the quaternary structure, and the hemoglobin usually is used as an example, and the red circle here stands for the heme molecule, which contains the oxygen, and you know that hemoglobin transfers the oxygen from the lungs to in the blood to the different tissues. Okay. So this summary of the structure of the protein. The primary structure is related to the linear arrangement of the different amino acids in the polypeptide chain, and usually the word linear gives the impression that it is a straight line, but it means that there is no any branching. It cannot be, not necessarily always to be like a straight line, but is linear joining between the different type of amino acids, and this form the skeleton or the backbone of the polypeptide chain of the protein. And then, according to the nature of the amino acids in this primary structure, it can have some sulfur bridges in this folding or this folding, or it can have some type of again sulfur bridges here. And this will form either alpha helix in this shape, the secondary structure of the protein, within the same polypeptide chain between the first amino acid and the fourth or the third. So, for example, in this case, this is amino acid number 1, and this is amino acid number 2. So between this number 2, 3, 4, 5, and 6. So the fourth of number 2, they have solid hydrogen bond. And between this third amino acid, and it is with another type. The dots, the red dot, indicate hydrogen bond. So this alpha helix is between the same amino acid of the same polypeptide chain. The beta sheets is between two adjacent amino acid groups or polypeptide chain, and again, this is hydrogen bonds between the functional groups. This is a composite of this amino acid, and this is a amine of the other amino acid in the other chain. So this makes the secondary structure of the protein, and then the combination of some alpha helix with some beta sheets will give the tertiary structure of the protein. Finally, the quaternary structure, where you have got multiple polypeptide chains, each with its three-dimensional structure. They come together and they form non-covalent bonds, holding and stabilizing the relationship to do a specific function, like the case of the hemoglobin. Any questions, Shabab? Isra? Do you want to ask anything?
No.
Okay. Shall I continue?
Yes.
Regarding the structure of the protein, the levels is clear? Okay.
Yes.
Let's go to the classification. As we said in the introduction, the proteins are most abundant biomolecules in the living organisms in general, whether plants, animals, or other living organisms. And within the human body, again, they are the most abundant type of the biomolecules or macromolecules. They can be classified according to many aspects. So in this case, in the first classification, the general classification according to the simplicity or the complexity of the polypeptide chain. They can be classified into simple proteins, and this is when you break down the protein, it will give only a series of amino acids, nothing else, or it is formed up of only amino acid, polymers of amino acid only. The conjugated protein, sometimes you can find it as a complex protein, where it contains some other components like protein plus prosthetic group. It can be carbohydrate residue, for example, and in this case will be known as glycoprotein, or it can be a part of a lipid, it could be lipoprotein, and so on. Or derived or modified proteins, like the case of the modification of the amino acid. It will be modified by some means of physical or chemical means. It will be changed slightly from its original nature. So this is the general classification according to the nature of the protein, whether it's originally just pure protein symbol, only amino acid polypeptide chains, or it has contains some other residues or some other prosthetic groups like glyco residue or some protein, lipid residue, and so on. Or it has been modified or changed in some of its parts by some modification. The other way of classifying the proteins is the general structure or the shape. The proteins can be classified mainly into either globular proteins, and these are of like a spherical shape in general. And you may know that the protein, they have not typical structure as which where we can say spherical, but it has like this globular shape because the general orientation gives you the impression that this is look like a spherical orientation of the protein or polypeptide chain in its three-dimensional structure, or fibrous protein. And the main difference between these two classes of proteins, the globular proteins are usually known as the plasma proteins, or the plasma proteins are of the globular nature or structure or shape, and the fibrous protein are related to the skeletal, the structural proteins like the collagen, elastin, keratin, and the other examples of the building or structural proteins. Again, the globular protein, because they are mainly plasma protein, they contain polypeptide chains of amino acids which are hydrophilic. They contain functional groups that can be water soluble because they need to be dissolved in the plasma and transported in the plasma from the original synthesis site to the site of the use, or site where it will do its function. The fibrous protein are mainly from the polypeptide chain where they contain hydrophobic amino acids. And this hydrophilic and hydrophobic classification of the amino acids, if you remember, it depends according to the R side chain, whether it contains some hydrophilic nature or hydrophobic nature. The other way of classifying the protein is according to the functions that the protein plays. And this is a summary of the classification according to the function. So you can find proteins playing a catalytic function, accelerating the reaction, the rate of the reaction of the biological system, and these are known as enzymes. And you know all enzymes almost are proteins. You can find some few enzymes not protein, but almost all the enzymes are from proteinous origin. So their function is catalyzing or reducing the activation energy or accelerating the action. So catalytic process or function. Structural proteins that are part of the structure of the cell or the tissue, and this is example of this is the collagen, which is found in the muscles and the different structure of the body. The contractile proteins, the actin and the myosin of the muscles, are the example of the proteins which form the contraction and relaxation of the different muscles, and this relates to the motion of the body, especially the muscles. The natural defense proteins, the immunity proteins, the immunoglobulins or the antibodies are mainly proteins, as you know, and they are formed mainly in the liver or sometimes in the lymphocytes. Transport protein, like the albumin, globulin, hemoglobin, all these are transport protein, which they can carry the non-water-soluble molecules. Like, for example, the albumin carries a free fatty acid in the bloodstream from the site of their storage in the adipose tissues, for example, to the other tissues where it can be used as a form of energy source in the muscles, for example, or in the heart or any other position. The globulin, the hemoglobin, which carries oxygen and so on. The blood protein, these are the clotting factors. The fibrinogen is an example, and most of the clotting factors are type of proteins which are synthesized mainly in the liver. The hormonal proteins, example of this is insulin, or the respiratory protein, the cytochrome group, can be cytochrome C, cytochrome B, 450, and so on. Also, the repressor proteins, these are the gene and the expression regulator of the genetic code and the genetic process of the regulation of the protein synthesis. And the receptor proteins in the cell surface, some proteins are forming the receptor of the cells, where they can give the information to the cell whether to accept the individual molecules, ions, or nutrients or not. The ribosomal proteins associated with the protein synthesis, where the transcription takes place, and the polypeptide nucleotide will be transformed into the amino acid synthesis or the primary structure of the protein. The toxin proteins, like the bacterial toxins, as we mentioned in the first slide of the introduction, or that you can have some type of the proteins or the amino acid in the form of plant toxins or bacterial toxins. Finally, the denaturation of the protein, as we mentioned, the normal structure of the protein has got a unique three-dimensional structure. This is an example for any protein. If it loses all this structure by any means, this can include some change in the pH, or the temperature of the medium, or the ionic strength of any heavy metals, or the solubility in the fluid or the liquid where it is present, whether in the lab or the tissue, it will lose its three-dimensional structure. So it will be denatured protein, and it will no longer do the same function, although the individual polypeptide chain of the protein is as it is. The number of amino acids are the same, and the primary structures, the bond the peptide bonds between the different part of the polypeptide chain are the same, but because it is loose or has lost its three-dimensional structure, it's no longer that specific protein. So this is exactly imagine this is albumin, which will take the free fatty acid, and by any of these factors, pH change or temperature increase or decrease in a very significant way, or it has been exposed to some ionic states, like for example heavy metals, for example lead or copper or any of the heavy metals, or the change in the solubility. Lost, it has loses its three-dimensional structure, so it's no longer a polypeptide chain of the three-dimensional structure. It will not do the nature of the protein albumin. Removal of any of these factors can take the protein back to its three-dimensional structure, and this process is known as denaturation. So the denaturation, by losing the biological activity due to any of these factors, and removal of the factor or the effector factor, it will take the protein back to its nature. This is another example where you have got two polypeptide chain, A and B. They form this native nature of the protein, and because of any of these factors, it has lost its orientation of the three-dimensional structure. It became this denaturated protein, and the A and the B separated from each other and became like non-three-dimensional structure. And the renaturation needs to again remove the effector factor to take it back to nature. So, in summary, the three-dimensional structure of the protein is very important and is achieved by the four levels, namely the primary structure, where we mentioned it related to the skeleton of the polypeptide chain of the amino acids, where it's linked by either peptide bonds or sulfur disulfide bonds or sulfur bridges. And the secondary structure, where you can form the alpha helix or beta sheet either between the same polypeptide chain or the two adjacent chains, and the tertiary structure of the protein, where we can have the different secondary structure alpha helix and beta sheets combined to each other by some forces and covalent bonds, forming the three-dimensional structure. Some oligomeric aggregation of proteins can take place. For example, for the hemoglobin, where quaternary structure of the protein is achieved by four different subunits, each with three-dimensional structure. They form non-covalent bonding between each other, and they form the aggregation of the protein to fulfill the transfer of the oxygen in the blood. The bonds for the primary structure is peptide bond, and this is the covalent bond. You can find also the sulfur bridges or the disulfide bond against covalent bond, while the other levels, whether from the secondary, tertiary, or quaternary, is non-covalent bonds. A single amino acid substitution can give rise to malfunctioning protein, and subsequently, if the substitution or the deletion or the addition of more than one amino acid, for sure it will be completely with different functions, and it will give malfunctioning of the protein, or it will destroy the function of the protein. Thank you for your listening and your time. Any questions, Shabab? Any question? أي سؤال؟
واضح يا دكتور.
شكرا جزيلا. ما انا ما شايف باقي السكرين، لانه اول يوم الليله درس في المنصه دي. الحضور اللي ما زالوا هم نفس 9 ولا زادوا او نقصوا؟
كان وصلوا 30.
اوه كويس، جميل. على كل انا يعني متاسف جدا لو في اي يعني inconvenience في طريقه البدايه بتاعت الليكشر او اثناء التدريس، لانه كنت في الكونفيجن بتاع كيف تستارت المنصه وكده. وأتمنى إنه اللوجو بتاع جامعة بيشا والصفحة الأولى إذا في طريقة في أجهزتكم تنزلوها فقط لحق النشر بتاع الجامعة والكوبي رايت بتاعت الجامعة. وفي المرات الجاية إذا درستكم إن شاء الله حعمل لكم سلايدات خاصة بكم أنتم ومن الأهداف التعليمية المذكورة في البرنامج. لكن بصورة عامة في كل الجامعات البروتين ستراكشر بتدرس على المستويات دي. كنت حاسس إنه ربما يكون في ديفيشنسي في جزء على امينو اسيد وطبيعتها والكلاسيفيكيشن بتاعها والبيبتايدز والانواع والنيتشر بتاعت البوليبتايد تشينز و200 تسمى بروتينز. لكن من بعض تفاعلاتكم اللي حصل انه اخذتوا بها حسيت انه الناس اتليست اورينتد. ربما اخذتوا الكلام ده في محاضرات جوجل وربنا يجعل الحرب دي. واسال الله سبحانه وتعالى انه يرفع البلاء ويكشف الغمه عن الامه السودانيه كلها. اي سؤال يا شباب وانا اقفل السيشن؟ Any question? أي سؤال يا شباب؟ شكرا لكم يا شباب جميعا ونلتقي في مرة أخرى. إذا عندكم أي هنا الرقم بتاعي مع الأستاذ طارق تقريبا كان عندي كوميونيكيتر ستيور برزنتيشن. إذا حبيتوا تتواصلوا معي بأي سؤال في المحاضرة دي أو إذا جات فرصة لمحاضرات أخرى يعني I'm ready to respond to your questions at any time. Thank you.